Hi, everybody. Um, Welcome to our webinar, A New Turn in Peptide Purification. My name is Eric, and I will guide you through this presentation as your moderator. First, we would like to talk a bit about Bachem, as we are hosting this webinar. Bachem is a leading innovation-driven company, and we specialize in the development and manufacture of peptides and oligonucleotides. We have now 50 years experience in the business, and we are experts in production of research, clinical development, and commercial peptides for pharma and biotech companies worldwide. And we have a whole other range of services as well. We operate internationally, and we have our headquarters in Switzerland and locations in Europe, the US, and Asia. We are stock listed, and for further information, you can see our homepage. First, a bit about myself. As I already said, my name is Eric. I'm a sales manager for custom synthesis at Bachem. I'm located at our site in Switzerland. I'm the primary contact for our partners from Germany, Scandinavia, and Asia in the area of R&D, diagnostic, and cosmetic peptides. I support our customers all the way through the project, from the first inquiry until everything is shipped. And I've been with the company for 2.5 years now. So now we would like to get to know you a bit better. And now, in a second, a question will pop up on your screen where you can tell us if you already have some experience with peptide purification. We would like to get to know you a bit better and to know what we should focus on in our talk, and just in general are interested in what you experience, what experiences you've had so far with peptide purification. Has it been going well? Did you not do it so far, or did you had some troubles before? So questions and answers are running in right now, and we should see the result soon. Yes, there's the result. And we see that we have a proper audience here, namely that about half of you have experienced some trouble already, and we hope that we can help you with the catch and release technology that we're going to present to you right now. Thank you very much. So yeah, a bit of a short overview what we can all offer at Bachem so far. So we can support you in every stage of the development of a peptide process or oligonucleotide project. We provide services for research and development, for clinical applications, and for commercial applications. So we can really go from the first milligram to the hopefully large-scale commercial supply that you will need for your peptide once it is on the market. How do we do that? We have a number of sites worldwide. As I already said, we are headquartered in Switzerland at our site in Bubendorf. We have a second site in Vionnaz, where we produce up to ton scale small molecule APIs. We have 2 GMP production sites in the US. We have a small sales office in Japan. And what we are especially proud of, we have our non-GMP production site in St. Helens in the UK, where actually a lot of the work that we're going to present to you today has been performed. And we're really happy to present these results to you. Before we're going to start, short information. Please be aware your camera is turned off. You cannot turn it on, but also don't be afraid. We cannot hear or see you. This is as designed. If you have any questions, please write to us in the Q&A, and we will try to answer these questions after the presentation in the question and answer session. If you need immediate assistance, you can also type to us in the chat, and we'll try to get to you as soon as possible. Now, I would like to quickly introduce our speakers. From Bachem UK, it's Dr. Gavin Noble. He's a research and development chemist at Bachem UK. He's primarily undertaking investigations into new production measures for research-grade production, as well as supporting our synthesis and purification groups if they run into any troubles. He's been with Bachem since 2014, and Before that, he was a postdoc at the University of Notre Dame in the USA, where he researched peptide-targeted liposome nanoparticles for cancer and antimalarial therapies. Prior to this, he obtained his PhD from the University of Manchester in the UK, where he performed research into protein and enzyme activity at synthetic liposome surfaces. He has published several peer-reviewed articles in high-impact journals with over 600 citations. And from BELINTEC, we are very happy that Dr. Robert Zitterbart is joining us. He's the co-founder and head of R&D at BELINTEC in Berlin. He's focusing on developing linker systems to purify and modify peptides. And he has almost a decade of experience in the development of peptide purification technologies. After his diploma work in the group of Peter Vollhardt at the University of California in Berkeley, he earned his PhD at the Humboldt University in Berlin in the group of Oliver Seitz. He has also successfully published many peer-reviewed articles with a lot of citations. So we're really happy that we have two experts on the field joining us today for this talk. A short insight in the collaboration between BELINTEC and Bachem. BELINTEC is a startup in Germany with a strong expertise and a high innovative potential. And Bachem was very happy that we could enter into a sustainable partnership right from the beginning and focus together on this innovative technology, because we saw a huge potential to purification of peptide APIs, which would really serve our customers. Bachem has a strong expertise in the development and large-scale production of APIs, and we're really happy that we can collaborate with BELINTEC on this technology. Just a short overview what we're going to do next. Robert will start. He will give you a short insight about BELINTEC, the general principles of the catch and release technology, and then the application to peptide sets in the parallel purification. Afterwards, Gavin will take over. He will give a short insight about Bachem UK, the advantages and limitations of a base labile linker, and the working principle of the newly developed reductively cleavable linker system. And in the end, we will have some time for questions and answers. With that, I'm happy to hand over to Robert and his part of the presentation.
Yeah, thank you, Eric, for the kind introduction. And I hope you can hear and see me well. And welcome to my talk about BELINTEC's Peptide Easy Clean technology. Let's see if I can. Yeah, okay. So who are we? As BELINTEC, we are an independent chemistry for healthcare enterprise which was founded in May 2018. And as Eric already mentioned, we are in Germany, so more particular in Berlin. And we have a strong background in peptide and organic chemistry. The current offer of our technology, that is a catch and release based technology, builds on kit products and service offers, as I will explain to you on the next slides. Yeah, so the services that we are providing include an implementation service, linker molecule design for particular needs, protocol development, also project collaboration. The kit products we are currently offering have 3 different categories. So the first one is research kits. They are ready to use. So they come with this pre-packed cartridges and all chemicals that are needed to do our pack purification. So they have 3 different sizes to whether purify 24 peptides or 8 peptides and 25 micromole or 100 micromole scale. And then we have the developer kit. This is more bulk material kind of kit, where you just get the linker and the filter material, but you can then create any formatation of this. So you can whether purify many peptides in small scale or one particular peptide in a large scale. And additionally, we also offer high throughput kits in two sizes where you can whether purify 48 or 96 peptides at a time. And I will show you some examples. So the PEG technology enables rapid parallel purification. So why parallel? So why do we need many peptides rapidly? Well, for example, all of you know now this guy here, the coronavirus or SARS-CoV-2 virus. And if you want to investigate it or if you want to fight it, for example, there are also peptide therapeutics and also peptide vaccines under development against the virus. But also if you want to understand its function, for example, why the B117, the UK variant, is more transmitting than the other variants, you can test the peptides that are altered in the mutations and estimate what causes this. So peptides are useful, and when you, for example, investigate this virus, you have to synthesize many peptides. But first, how does the technology works? So I'm not going into much detail here. So it's all built on this PEG linker. It's a small molecule and it's reductively cleavable. We designed it to be side reaction free, and it's patent pending, and it's available in kilogram scale from this year on. It features three main functionalities. So one functionality is this Boc protected oxyamine here as a catch tag for immobilization of your tagged peptide. And then, very importantly, you have a cleavable core here, or cleavable unit. This is a aryl A site that can be reduced to release your peptide. And here, it's a leaving group to connect the linker to your desired peptide. Gavin, in his talk, will go into more details and more chemical details, but here's a general overview. So the technology is built on the solid phase peptide synthesis, so SPPS, where you assemble amino acids after each other on the polymeric resin. So the amino acids are here, this purple circles. So, and what you usually experience is that you have some truncations building up during the synthesis that you can block away by capping, usually with acetic anhydride here, represented as a red square. And then after you're done with your SPPS, you can couple the linker as a last residue to your desired peptide. Here's this green circle. So it's exclusively coupled to the full length peptide, and there is the separation happening already. And then you cleave off your peptide of interest with all the crude material inside, also solvents, not only truncations. And then you can dissolve it, and only your desired peptide is caught on the purification media, and the rest you can wash away. And then you apply a chemical stimulus, in our case a reductive agent, and thus releasing the peptide. So the filter material we are currently using and providing is activated agarose, which is highly stable and has a high loading capacity. So how does this really work? So here's an example of a neoantigen set. So here's a little bit a picture of how this looks. You can also follow this link later if you get the presentation, or if you watch it on YouTube. The link will be also be in the video description. And then you can watch a video how it's currently working. The process currently is manually, but we are also working on automating it. So it starts again by the SPPS synthesis on a synthesizer, where the linker can be coupled on the synthesizer. For example, this particular peptide from this neoantigen set has been performed a linker off crude analysis, and purity of 42% was determined. After linker coupling, the retention time of the desired product shifts a little bit to higher retention times. And then when you have this crude product where the peptide is linker tagged with a linker, you put it on the agarose material in this cartridges, for example, if you use a research kit, and then it just vanishes from the supernatant. You can analyze a supernatant by UPLCMS, and then you see the desired peptide vanishes and all the dirt stays in the supernatant. Then you wash, you cleave the linker, and thus releasing your purified peptide, in this case, gaining 91% purity. In this particular peptide set, you can read all the details here in our publication together with Bachem in Chemical Science. The crude purity was improved from 53 to 89%, and we had a decent recovery of 75%. And it was all, even though the peptide showed a high variety of parameters, it was generally applicable. So it was really a one-size-fits-all solution. And the required time was just 3 hours active time and 3 hours passive time, waiting time. So, but what to do when you need even more peptides? Then you can use the high throughput purification in plate format in this high throughput manner. So again, you start by the synthesis. Again, linker coupling can be done on a synthesizer, and then you do this washing and handling in a plate format. And the vacuum manifold one can use is shown here. And then you get your lyophilized final product also in a plate. Don't wonder here, we didn't purify all the 96 peptides, but just 56, I think. And here's an example that we got together in the development phase, together with the working group of Professor Stephen Kopp, where we are allowed to show sequences. So for example, this was a crude purity here, a wide distribution of purities. And after the PEG purification, we had a mean purity of 88% and a good range of purities ranging between 74 and 97% purity. So here in the Corona context, we also purified for customers other libraries, and the outcome was even better. And all those peptides have been performed very well in biological assays, as is shown here in a T-cell stimulation assay. And yeah, I want to show you this picture. This is actually the cover picture from the Chemical Science paper. And I want to convey the notion that PEG can be seen as a peptide dishwasher for parallel peptide purification. Okay, thank you very much for your kind attention and looking forward to your questions.
Okay, oops. Thank you, Robert, for your talk, and thank you, Eric, for introduction earlier. My name's Gavin Noble, research and development chemist at Bachem UK, and for my part of the talk, I'll discuss the development work that we put into forming the new reductively cleavable linker over the past few years. But first, I'll just give a quick introduction of St. Helens site for Bachem UK. So just a bit of context on why our site was a nice fit for this kind of technology. So we're the Centre of Excellence for Peptide Manufacturing, based in St. Helens, which is in the northwest of England, quite close to Liverpool and Manchester, if you know that part of the world. We are ISO 13485 certified, which is ISO certification for medical devices. Recently recertified this year as well, so we're doing well. This system allows us to fulfill several of Bachem's key product lines, those being research chemicals, custom synthesis, and we also supply several of Bachem's products for their online catalog, in addition to our growing work with our diagnostic partners. In order to fulfill this, we have very rapid and robust production methods. As you might expect, we have a suite of automated peptide synthesizers and purification equipment that allow us to produce over 1,000 peptides per year, over 1,000 different peptides per year. Last year we were actually synthesized 1,400 peptides. We also are capable of a wide range of unusual peptide modifications, anything from fluorescent labeling to stapling. Yeah, anything you can think of, really. And despite these challenges, we are still capable of producing peptides in really high purity. Standard of 95% is typical. I should also mention our typical scale ranges from the milligram to the multigram scale. So with that in mind, we very much service the research and specialties part of the Bachem 360-degree business model, but also we have some exchange with other Bachem sites, which enables CMC development for projects going forward. So with that in mind, we had some initial meetings with Belintic when the project kicked off. So we wanted to get on the same page with regards to our expectations and potential uses of this technology, and also align our analysis and peptide production tools, just to allow consistency across sites. So the state of play when we started the collaboration a good few years now is the base labile linker. So this linker, after SPPS, is coupled to the N-terminal of the peptide. As Robert mentioned, the para-nitrophenol leaving group is employed. It's simply mixed with oxymer and DIA, and then is appended to the N-terminus of the peptide while on resin, selectively to the full-length peptide. After washing and drying, the peptide is cleaved in the normal manner using TFA and regular scavengers for cleavage. The system is robust towards several common scavengers such as EDT, TIPS, DODT. Tried several already and it's fine. Once you have obtained your linker modified peptide and then immobilized it, the basic labile linker is removed or released from the agarose resin using amine. In this case, it's methylamine. So as you can see, that the release is really rapid and typically is most of the peptides is off in 60 minutes. So in order to support the collaboration, what Bachem UK did was synthesized a group of peptides that are representative of the kind of work that we do at Bachem UK, and maybe peptides that were relevant to this kind of technology, or where this technology would benefit. So what we selected is a mixture of hydrophobic, some of our catalog peptides which we've made, and also some difficult peptides with a representative length of 20 amino acids. These are all synthesized by ourselves at Bachem UK at the millimole scale, and we split the syntheses between us, and half of it was shipped to Belintic and half kept on site for simultaneous evaluation at both sites. What we did see is a really good purification purity increase across the board, aside from a few inconsistencies. So if I draw your attention, the amyloid peptide, particularly hydrophobic peptides, in this case, there was a drop in purity after catch and release, but this we found was due to solubility issues, subsequently solved, I should say, as well. But several of the peptides showed significant decrease in purity, which were not as a result of handling issues. And yeah, this compromised the purity, and we wanted to know why. Okay, so despite the setbacks from certain peptides, what we did see is for some peptides, the technology could be really, really handy. For example, this histone peptide, which is very hydrophobic and early eluting on LC systems, so it can be very difficult to purify. I mean, what we saw is that the crude chromatograph looks really nice, but underneath the main peak are several capped impurities. And when we performed catch and release on this peptide, all of these impurities can be chemoselectively removed. And yeah, it's rapid term over time, so this can all be done within several hours, as Robert said. And this gave a purity of 81%. Interestingly, if we apply both catch and release and traditional LC methods, so prep LC in this case, we can achieve super high purity, which shows the benefit of the two orthogonal methods working hand in hand together. So as an aside, it took 3 rounds of prep LC with multi-dimensions to achieve this level of purity alone. So that, again, it demonstrated the potential for an orthogonal approach for particularly tricky peptides to purify. And this can be seen on the case studies on the Belintic website, some of our collaborations there. So yeah, despite the advantages, the disadvantages remained for the side reactions, and we wanted to go back to the drawing board and develop a new linker that was capable of overcoming these induced side reactions. So the Belintic gang developed a new reductively cleavable linker for a milder release and a universally applicable release step. So the new linker retains the oxyamine catch tag, BOC protected as well, and also the paranitrophenol leaving group for appending onto your N-terminus. But in this case, there's a para-azidobenzyl cleavable unit, which is sensitive towards reducing agents such as triphenylphosphine or diethiothriatol. So in this case, the reductive cleavage is undertaken in 3 steps. So using reducing agents to reduce the azide to an amine, washing out any excess reducing agents, and then the final step is treatment with aqueous acid, which causes a cascade, leaving the peptide behind. So going into more chemical detail, we can follow this workflow through. So SPPS is performed as normal. TFA cleavage, as I mentioned, for the base label linker, again, this linker is not sensitive towards any TFA cocktail scavengers. And after you've obtained your crude peptide labeled with the linker, you immobilize on the agarose resin. Typical buffer of choice is DMSO to solubilize the peptide, diluted with citrate buffer. But the loading step is also very tolerant to many organic solvents, as DMSO, DMF, HFIP, as to name a few. Once the peptide is immobilized on the resin and then washing is being performed to remove any truncated impurities, the resin is then treated with triphenylphosphine to form this aminophosphorane intermediate. Then excess triphenylphosphine is washed away and the resin treated with aqueous acid for 90 minutes to induce a 1,6 elimination cascade. So this releases the free peptide and the linker remains attached to the agarose resin, meaning that it's a traceless release. So then, yeah, after elution with TFA or other aqueous acid, ether precipitation allows the recovery of the full peptide, the purified peptide. Yeah, the ether precipitation step is necessary to remove any excess triphenylphosphine oxide. Alternatively, you can use other reducing agents such as DTT. And in this case, yeah, after immobilization, you mix with DTT with sodium bicarb to reduce the azide. This again can be washed to remove any excess reducing agents, and then acidic cleavage affords the peptide through 1,6 elimination. An interesting feature of this, and for triphenylphosphines actually, is that the linker, the immobilized peptide is stable towards many species at this washing step. So it's stable towards basic and neutral conditions. And this aspect has been investigated in our labs. And so we went back again to our peptide panel and looked at the purification, again, trying to see if we had any side reactions this time. And what was great is that we saw no side reactions that were previously observed. And also importantly, no additional side reactions were observed. And this was evidenced by the increase in purity and the absence of any side reactions in LC or MS data. And for the histone or shorter histone sequence, we again saw consistent purity, so no decrease in purity for ones that worked well. However, we did see a decrease in the recovery of the peptides after catch and release. We initially thought that this might be due to incomplete coupling of the linker, but then further investigation showed that the reductively cleavable linker is actually unstable towards the TFA cleavage step. But again, back to the drawing board, and an addition of an electron withdrawing group to the azidobenzene ring stabilizes the linker towards TFA cleavage. So model compounds showed that the stability during TFA cleavage. So bromination at the X position increased the stability towards TFA and also allowed high purity as before. Interestingly, if you add a further bromyl group, bromination to this Y position to give 2C, you see decent purity, but also lower recovery. And this is due to the molecule actually being too stable towards reductive cleavage. So the sweet spot and the currently commercially available linker molecule is monobrominated at the X position, which gave both good purity and good recovery for all the peptides tested. And again, to reiterate, the no side reactions occurring. So with all this in mind, we have a really robust, widely applicable system that can be deployed for rapid parallel purification of peptide libraries for API development and other peptide developments. We developed the first-in-class reductively cleavable linker, and it was shown to be really, really useful for purification of complex peptide mixtures, such as the histone peptide shown. The linker is currently available for research grade purification within Bachem as well. So yeah, if you have any projects in mind with these, where this would be useful, please let us know. And with that, I will hand back over to Eric for another poll.
Thank you very much, Gavin. So I hope we could convince you that the Peptide Easy Clean technology is a valid addition to your repertoire. And we would like actually to test this right now and ask you, do you think the Peptide Easy Clean technology could help you in your projects? Please let us know what you think about it and give us your honest feedback so that we can see immediately how well we explained this to you right now. Again, I think some answers are coming in already. Let's just wait a bit longer. And the results are coming in. Very nice. So we did a good job. If you need more information, like the 20% of you that answered that, please either post a question right now in the Q&A session or just contact us and we'll get back to you. So now the next step would be to proceed to the questions and answers. I would like to start with the first one. So yeah, Gavin and Robert are coming on the screen right now, hopefully. But yeah, let's start. I think, I don't know, Robert, are you there?
Yes, I'm here.
Yes, you are. Perfect. I just wanted to make, because the first one is for you. This says, hi, first of all, amazing technology. I was wondering if capping is necessary after every coupling to prevent residual activity on the linker.
Yes, I'm not 100% sure what is meant by residual reactivity, but so the capping is not really necessary to tweak the reactivity of the linker. It's just to prevent the truncations from reacting with the linker, because eventually after the TFA cleavage you will fish out everything that bears the linker, and to make sure that only the desired peptide bears the linker, you can do capping. So usually we recommend to use capping if you don't know much about your peptides. So if you, for example, if you synthesize a peptide library of 96 peptides, I would just recommend to cap everything, and then you have a high chance of having a good result. But if you think about API synthesis, for example, you don't necessarily need to cap after each step when you know at what junction you have poor coupling results. For example, you synthesize a 20-mer, let's say, and after the 15th amino acid, your coupling efficiency goes down. It's okay to just cap after the 15th amino acid if your coupling efficiency before that was always 99.9%. So that's feasible. And the linker will, for example, in this histone case that Gavin showed, it's also quite aggregating the peptide, therefore difficult to purify. And in this particular case, we didn't cap so efficiently, but if the capping is not efficient enough, the linker will also not couple with this truncated sequence.
Thank you. Very good. We have another question, and this is also for Robert, and this is, does Peptide Easy Clean have a limit in length? Peptide length, of course.
No, actually, we think that as longer the peptides get, as more you can benefit from the technology, because the likelihood of that the truncations will build up is increasing with length. For example, we have a case study on our homepage you can download showcasing a 84-mer, where the peptide of interest is basically or sits on a big hill of truncations, and this would be by traditional chromatographic means quite difficult to be purified. but PEG enables it to just be pulled out of this dirt. And then, as Gavin also said, then you could, if you're not happy with this purity, at least you have a really good purity, and then you can polish it by HPLC to get from 80%. That's what we usually almost all peptides are above 80% purity. But if you want to reach then always over 90 or 95, then sometimes you have to polish.
Very good.
It's a really nice tool to have in your toolbox for peptide purification.
Thanks.
Great. We have another question, and this is, could you please comment on the compatibility with sidechain amines like lysines in this technology?
Yeah, this is feasible. So we have tested this. Also, customers have done it. It would call for an orthogonal protecting group. For example, what has been done is so maybe one sentence more. Unfortunately, we cannot, with our technology, or one cannot purify peptides when the N-terminus is modified or acetylated. But if you have the linker on the lysine side chain, it also works to go to purify over the lysine side chain. And how this works is, for example, you can use a lysine MTT or MMT, and then you deprotect it with 1% of TFA and DCM, and then you couple the linker to it, and it works just fine. Also, what has been also done is DDE protected lysine. So you deprotect the DDE with hydrogen, for example, and then afterwards you couple the linker. That works.
Great. Thank you. We actually have another question regarding lysine sidechain, and this is, are the lysine sidechains also reacting with the aldehyde on the beads?
It's difficult to answer this, so usually not, but sometimes they do, especially when you have a lysine close to the N-terminus or at the N-terminus. What happens is that the linker first, the amino oxy moiety reacts with the aldehyde, and then a neighboring amine group from the lysine may react with a neighboring aldehyde group. So you have like a multivalency effect. But this is prevented, or we do after the immobilization, we do a cysteine capping that reverses the lysine imine formation. So it's detaching the lysines again, basically, from the agarose.
Thank you. We have another question that goes, I think, in the direction of antibody-drug conjugates. And the question is it possible that the self-immolating Wald-Cit PAB linker is leveraged with this technology as well?
I'm not so familiar with this particular linker. If it is also reductively cleavable and it's also a self-immolative 1,6 elimination linker, then it may be a problem. I try to Google this.
I think it's enzymatically cleavable, Robert. So
yeah, so then it should be feasible.
Okay,
because it's orthogonal to the release methods from the catch and release.
Yeah, it's as far as I see, it's also a 1,6 elimination linker, but the trigger is different. In our case, the trigger is the reduction of the A site to the amine, or we have basically two trigger or a two-step release process. So first we need that the A site is reduced to the amine, and then the final trigger is the acid that is actually protonating the carbamate, thus releasing the linker. But this is actually just only feasible for the brominated versions of the linker. This enabled this acid switch, if you wish to call it like this. And this is enabling the DTT washout, because you reduce with DTT, you get from the A side the amine, and then it's not, or it's still stable, the linker. So the peptide is still attached to the resin, and you can wash out the excess of DTT. So for example, if you would use it together with this linker in whatever, in whatever way, what you could do is, if you want to cleave this this PAB linker first, you could add the enzyme first, and then it would release without touching the lytic linker or the PEG linker, or vice versa, that you first cleave off the peptide and that doesn't touch the PAB linker, and then you add the enzyme to cleave it.
Great. Sounds very good. I think we went through all the questions we had right now. If you're having additional questions, you can easily contact us afterwards. We have an extra email address set up for this, webinar@barcam.com, and we will then forward your questions to Robert and Gavin so that they can help you further, and also just contact us if you need more information material or if you have general questions. As always, please be aware we are on social media, so is Belintic, so please have a look there and see whether you can see something that you like. Thank you very much for your attendance and have a great day.